Okay, so we just went over Mendelian genetics. Now we are going to go over all the different ways we can break the rules for the Mendelian genetics. So that's what happens when you study something too much. You know, you start finding ways around it and some things that might not follow those rules. So that's what we're gonna do next. So based on a dihybrid cross, there was a law of independent assortment, and we went over this last time too. So the law of independent assortment says there's one gene on each chromosome. During meiosis, they mix, and then by random chance, there are different alternatives for each. Let's say these are the sperms, and there's a 1 in 8 million chance, we did the calculation for that, that this sperm is going to have big R, big G. Same thing for this one, 1 in 8 million chance, it's going to have little r, little g. So this is the law of independent assortment. And this is called segregation of alleles. So you have different combinations of alleles for that gene. So because there's one gene located on each chromosome, it's not really influencing the other gene. So what I'm going to show you next is this. This is saying you can have more than one gene on each chromosome. So you have V, W, and Y. You have 3 genes on this chromosome. Here, you only have 1 gene on this chromosome. So if you have more than 1 gene on the chromosome, it's not going to follow the law of independent assortment. And if you guys remember crossing over is the mixture of the genetic material. If the genes are located really close to each other, they are less likely to cross over. It's hard for them to separate, basically. So if they're close together, crossing over is less likely to separate the alleles. If these are still far away, so there might be still some crossing over happening. So going back to when I said that for law of independent assortment, there's 1 gene on each chromosome. Here you have 2. Let's say that this gene is for your appendix. Do you guys know what appendix is? Anybody have their appendix removed? Nobody have their appendix removed? So appendix is this worm-like structure. It looks like a worm. And they don't know what the function of this appendix is. It has no function, basically, as what they've been saying. What's in here is basically you poop. So it's poop there. And if this appendix bursts, it can cause infection and eventually death if it's not taken care of. Anybody have it removed? Appendix, maybe? No? So nowadays, when they do the surgery, they have these tiny cameras and they just create a very little scar, really tiny scar, and they can remove it. But if it's emergency where it has burst, they might have to just go in and remove it, and then you might end up with a bigger scar. So this appendix is serving no function, but it can cause death if it bursts because the poop has a lot of bacteria that can cause infections. Now, our ancestors, if we're looking at maybe cow stomach anatomy, so our ancestors, maybe they were eating a lot of, plants, and cows, they have multiple stomachs, and that's because the cellulose in plants, it's hard to digest, so it has to go through multiple functions. So maybe before our ancestors, maybe they ate a lot of plants or plant material, and maybe we had more complicated digestive system, but then maybe we started eating meat and we were more omnivorous, and then it didn't serve any function after. So maybe let's say this gene is for appendix, and appendix is serving no function. and it's a leftover structure from our ancestors. That's called a vestigial structure. So vestigial is something that's left over from our ancestors, but it's not serving any function. But on top of that, it can burst and it can cause death. So it's not really good to have this gene, basically. That's what I'm trying to get at. But then you also have another gene on the same chromosome that's for eyeball. Eyeball is really, really important. It serves, really important function. You won't be able to see if you didn't have your eyeballs. So because they're located on the same chromosome, it's your body's not gonna get rid of the gene for the appendix because it also has to get rid of the gene for the eyeball. So if they're located on the same chromosome and the gene still sticks around as long as there's another gene that has a really important function. So you can what I'm trying to say is that you can have more than one gene on the same chromosome, and one of those genes might not even serve any function, whereas the other has really, really important function. So it's going to stick around. Natural selection is going to keep it around because it has a gene that's more important. So if you were to compare, the gene for appendix maybe can cause infection, but compared to that, your eyeball is really, really important, so it's going to stick around. So this does not follow the rules for Mendelian genetics. So it doesn't follow the rules for independent assortment because you have more than one gene on the chromosome. Any questions on that? No? So I'm going to give you a few other things that do not follow the rules for the Mendelian genetics. For the Mendelian genetics, we learned about the dominant allele and the recessive allele. Dominant allele, if it's present, it's going to take over. And recessive allele gets masked if it's present with the dominant allele. If you have two recessive alleles, then they are expressed. So the gene expression that does not follow the Mendelian genetics is incomplete dominance, codominance, and pleiotrophy, or pleiotrophy, however you want to say it. So incomplete dominance, I'm going to use this example here for that. It's snapdragons. So you have red flowers for snapdragons. Then you have white flowers for snapdragons. When these two mate, or when you have a hybrid, it looks pink. It's not red, it's not white. If it was following Mendelian genetics, then we would say that the red takes over. If it's dominant, it's gonna be red. If this one was dominant, it's gonna be white. But here you're getting mixture, you're getting pink. So you're not getting this one or this one. So it's not following Mendelian genetics. And this is called incomplete dominance. That means it's not fully expressed, neither the red or the white. So it gives you pink flowers, and that's the concept for incomplete dominance. That's incomplete dominance. Does that make sense to you guys? I have one example for this as well. So there's no gene that's dominant in this case. This is not dominant because it's not red. This is not dominant because it's not white. So let me give you one example for this. So in incomplete dominance, the heterozygous has an intermediate phenotype. So what does this mean? I'm gonna do an example. We'll come back to this example after I show you what's going on. I don't like the way they labeled it, so I'm gonna use different labels. So for incomplete dominance, I'm gonna say if you have big R, then you have red color. If you have little r, then white, but they have to be homozygous. So your book is saying R1 is red, R2 is white. I'm gonna just say big R gives you red, little r gives you white. And then you do a cross. You do a cross with big R, and little r. So let's look at the genotypes and phenotypes for that. So you do the cross. If you have big R here, that means that's the red one. Little r here, that's white. You do the cross, you get heterozygous. In this case, heterozygous is not red, even though, I wrote it as a big R. If it was Mendelian genetics, then it would be red, 100% red. But in this case, it's incomplete dominance. So I don't know if you guys remember, I said Greeks, they said if you mix red paint with blue paint, you're gonna get maybe purple paint. So that's what's happening here. So in this case, I said big R gives you red, little r gives you white. It's not gonna give you red. Big R, little r is not gonna give you red because we're not following Mendelian genetics. This is incomplete dominance. In this case, it's gonna be pink. So it gives you pink. Now, this is your F1 generation. And then I say, oh, cross F1 with F1. So you do another cross. Now for this cross, since this is your F1, you say big R, little r, big R, little r. So it's pink. This is also pink. That's what you're crossing. So for this one, you're going to get big R, big R, big R, little r, big R, little r, little r, little r. Now I want you guys to give me the genotypes and the phenotypes for this Punnett square. This is your F2. So the genotype and the phenotype. What is the genotype here? And what is the phenotype? So Kaylin said the genotype is 1 big R, 2 big R, little r, and then 1 little r, little r. What about the phenotype? Baron said 1 red, 2 pink, and then 1 white. That is correct. So you guys are following me. Any questions? And this phenomenon is called incomplete dominance. So this is incomplete dominance, which means none of the genes are taking over. You have mixture, maybe a little bit of red and a little bit of white. That gives you pink. So no dominance. This is not following Mendelian genetics. So on the quiz or the exam, I will tell you which rules to follow. So here, this cross that I just did, that's the one that's being shown here. So the parental one is not being shown, but your book uses R1 and R2 for this. If that makes more sense to you, you can go ahead and use that. But I like doing it this way with big R and little r. So on the quiz or the exam, I will tell you which rules to follow. I will say, oh, this uh, generation follows the rules for incomplete dominance. And then I give you maybe, pink flowers and I don't know, orange flowers, what you're gonna get or something like that. So I'll tell you in the question, this is incomplete dominance or the Mendelian genetics. For Mendelian genetics, you're gonna say homozygous recessive, homozygous dominant, or heterozygous, or something like that. So that's Mendelian genetics. You can still do a Punnett square, like Mendelian genetics, but it's not really following the rules for the Mendelian genetics. So that's the first thing, incomplete dominance. Next thing that I have is codominance. So this is saying the heterozygous pink because it receives half of the dose of the red pigment conferred by the dominant allele. So it just gets half of this. That's why it looks pink. So this is codominance. It's the second thing. So for the codominance, the best example I can give you is your blood type. So there is type A blood, type B blood, type A and B blood, and it says none, that's type O. So this part is referring to the molecules that are found on your red blood cells. If you have on your red blood cells, if you have the A proteins or A antibodies, whatever, however you learned it, you have type A blood. If you have B, then you have type B blood. If you have both, then it's type AB. If you have none, that's type O. So hopefully you guys know your blood type. In some countries, they put their blood types on their dating profile and they use the blood type to, I don't know, make a better match or something. So hopefully you guys know your blood type. Do you guys know what happens when a person with type A blood receives a type B blood? So yes, your body is going to reject it. It's going to think it's a maybe parasite or something foreign that's trying to attack your immune system, and it's gonna start clotting. It's gonna clot it, and that's not good. So they always check your blood type before they give you, if you need blood. So um, is the blood owed universal donor? And then this is the universal recipient. So you can receive either A, B, and then this can be given to all these types because there's no antibodies on this. So for codominance, there are two dominant alleles present, and that we have is here. So two dominant alleles are present, both proteins coded by those alleles will be represented on the phenotype. And that's when we say the person has a type AB blood. Type AB has both dominant alleles present. So genotype has A and B molecules, therefore the phenotype is type AB. So that's codominance. I in this case is representing no proteins. So when we do the problems today during the lab, there is a problem that's gonna go over the blood type, and that's when you're gonna write it as little I for type O. So I'll go over that again in the lab. So that's codominance. Let me show you one more example for this. So here you have a red fish and a blue fish, and this is the mixture or the hybrid that's produced from these two. In this, you get some patches that are fully red and some patches that are fully blue. So that means there are two dominant alleles, they're both present. If this was incomplete dominance, then maybe red and blue would give you a purple fish. So this is not incomplete, this is codominance. Does that make sense? Can you guys differentiate between incomplete dominance and codominance?
Yes, ma'am, for me.
Okay. Okay. Next I have is pleiotrophy. So for the codominance, you're going to use the example for the blood type, and for the incomplete dominance, I gave you the snapdragons example. For pleiotrophy, I'm gonna give you the example of sickle cell anemia. Try to remember that for the exam. So sickle cell anemia, pleiotrophy. Giselle, are you referring to incomplete dominance and codominance? Okay, so incomplete dominance, you are getting a mixture basically. So here, if this was incomplete dominance, you have red fish and a blue fish, that's going to give you purple fish. You're getting mixture. There's none of the alleles that's taking over. You're just getting maybe half a dose of the allele. In codominance, you're getting both. Both are being expressed. You are getting a little bit of red, and then you're getting a little bit of blue. So both are being shown. That's codominance. Does that make sense? Okay. So we have, I don't know what happened. We have pleiotrophy. It means one gene has multiple effects on the phenotype. In this example, you have a gene for a protein, and it can affect the biochemical pathway. You have a phenotype 1, where this gene is affecting, maybe catalyzing the reaction here, and maybe it's catalyzing the reaction here. But for another pathway, it's blocking the expression here. So it can have multiple effects. So gene encodes for a protein that catalyzes reaction in two biochemical pathways and blocks other. So a gene can have multiple effects. So let me show you an example of that. Uh, okay, so there's this single gene that's responsible for sickle cell anemia. And just to remind you guys, sickle cell is when your normal red blood cell shape turns in a sickle shape, with sickle cell anemia. So there's one gene that causes that. And if you have that single gene, it can have multiple effects, means it can cause blindness, it can cause liver failure, it can cause heart attack. So it can have multiple effects. Here's another example. So the ability of a single gene to have multiple phenotypic effects is saying breakdown of red blood cells, physical weakness, anemia, heart failure, pain, brain damage. So all those phenotypic effects is just result of a single gene. That's pleiotrophy. That's pleiotrophy, this here. So if this example doesn't make sense to you, try to think about sickle cell anemia.